Hi, everyone. My lab designs models, statistical and machine learning models to decode how genomes work, how they evolve, and how they break in disease. And I'm also lucky to be the head of an institute that does not only computational technology development, building models, but also wet lab technology development with people who are really pushing the envelope on what we can measure in cells. And I think these two types of technology development, while they're often done in different departments at universities, it's really neat to be in an institute where they're happening hand in hand and in a closed loop like I'm drawing here, because I think that predictive models can help improve the wet lab technologies and vice versa. And so what a big push of what we've been doing is a particular pairing of technologies, broadly speaking, models that deep learning models that are sequence based, meaning they take sequences as inputs. And then I'm calling them sequence to X or sequence to activity, meaning that they predict something from sequence. And then pairing that with genome editing, which is getting more precise and more high throughput as a way to generate data sets for training these models, to test model predictions, and then also use the models to figure out how to make the technologies better. So we're very excited about a suite of projects around this. And so I'm just going to give at the beginning here a very high level description of three research areas where this approach that I'm describing of closely pairing sequence-based machine learning with genome editing is starting to make a big impact in our collaborations. So I'll just describe them, and then I'm going to take one of them, which is the genetic basis for human diseases, and go into it in much more detail for the rest of the talk today. So, as I said, genome editing is getting more high throughput and more precise, but if we imagine trying to decode a genome as big as the human genome, and if we understand the fact that most variants don't operate independently, but they operate in conjunction with other genetic variants on the same haplotype, you can imagine that figuring out a sequence to function relationship in the wet lab through genome editing would result in trillions of possible experiments. And that number is not computationally feasible or not experimentally feasible right now. And therefore, our solution is to be able to predict the outcome of the genome editing experiments before doing them in the lab. Not that we would expect such a model to be perfectly accurate all the time, but by running computer genome editing, so running simulations of genome editing and what we predict based on the information we have right now, what we predict the outcomes would be, can help us better design the experiments to prioritize that experimental effort, not only towards like testing our top hits or the things we think look most exciting, but also checking places where we have less confidence in the predictions and trying to generate data sets that not only will find more discoveries, but will also and accelerate, the rate of knowledge generation, but will also be designed in a way that they'll help us build better models. So the first application of this general approach I just want to briefly talk about is in the area of cancer immune cell therapy. I'm sure everyone's aware of CAR T cells and other approaches that are being pursued right now to engineer the immune system through genome editing to make it more effective at killing cancers. For certain blood cancers, this works in some of the patients. For other patients, not as well. And for most solid tumors, not as well. And so there's a lot of interest in trying to figure out how to make better cancer-killing T cells. And one way to do that is to put different kinds of proteins on the outside of the T cells. You could also change the genome inside of the T cells so that they behave differently. They collaborate more with other T cells. They hone in on cancer better. They stick around when they find cancer more effectively. And so we have designed a closed loop experimental and computational platform to try to optimize the process of deciding what genes to knock out or overexpress in T cells to improve immune therapy. And that also includes trying to target regulatory elements in addition to like targeting the genes directly. The second area, very different. I'm just kind of giving you an overview of many projects. This one is attacking the problem of antimicrobial resistance and the fact that increasing number of bacteria do not respond to the available antibiotics. And this is a major public health problem, which is predicted to become a top killer by the middle of this century globally. As you are also probably aware, bacteria have, there are natural killers of bacteria called bacteriophage or phage. These are the viruses that infect bacteria. They are different than the viruses in many ways from the viruses that infect human cells, but they are really interesting from a therapeutic perspective because they're pretty specific. Most phage only can infect and kill certain bacteria. So in that sense, they're much more narrow spectrum compared to, say, a broad spectrum antibiotic that'll wipe out a pathogen, but also wipe out a bunch of good microbes in your human gut microbiome, or microbiomes in other parts of your body. So there's been interest in phage for many decades, but the approach that people have been using is when a patient has an infection that is not responding to antibiotics, their bacteria is taken out of their body, cultured, grown in the lab, and then exposed to a collection of phages that people have pulled out of wastewater treatment and ponds and other environmental sites in hopes that somewhere in the collection there's a phage that can kill their bacteria. And if you find one that kills in the lab in the dish, then you try it in the patient. Unfortunately, this trial and error process rarely succeeds. There's some amazing stories where it has succeeded and saved people's lives, showing that there's a lot of promise. But there isn't right now sort of a rational way to design a phage therapy to look at the bacterial genome and the phage genome and predict whether a phage would be successful. So the approach that we're taking isn't trial and error. It's to try to build up that understanding of which phage can kill which bacteria, using data sets that are available from experiments, testing them against each other, as well as what we see in nature through metagenomic sequencing, which phage co-occur with which bacteria across lots of environments, and which phage are seen to have inserted into the genomes of different bacteria. All of that information build up a machine learning framework to predict not which existing phage, but maybe even a phage we've never seen before. What combination of phage genes do you need in order to kill a certain bacteria based on the genes in the bacteria's genome? And then be able to engineer those phage on the fly. And this would also be faster. Right now, the trial and error and culturing takes a very long time. This would be a 24-hour sequencing experiment of the bacteria, and then you could synthetically design these phage and reboot them and deliver them to a patient very rapidly. So I'm excited about the promise here. It's very early days, but I think that this will be a very nice example of this interplay between building a model and doing these genome editing experiments. And then finally, what I'm going to focus on for the rest of the talk is establishing causality for human genetic diseases. We now have the tools to go in and make all kinds of different edits in human cells and model genetic diseases. We can knock out genes, we can knock out regulatory elements, we can epigenetically up or down regulate them. We can even now, with tools like the Editron from Seth Shipman's lab here at Gladstone, go in and change single nucleotides. People are probably familiar with prime editing and other types of base editing. This expands that because it provides a DNA, provides synthesis of the repair template. So what Seth has done is hook up some bacterial retron, which synthesizes, it's part of the bacterial immune system, and it synthesizes oligos inside of bacterial cells in order to fight viruses. That's what it naturally does in bacteria. We're using it in human cells to synthesize a repair template. And then we hook that up to the Cas protein. So Cas with a guide RNA will go to a site and cut it. and then the retron will synthesize the repair template. This makes it much more efficient than other types of base editing, and the repair template can be several kilobases long. And so rather than making one point mutation at a time in an iterative round of experiments with cloning, here in a single step, we can replace a whole piece of the genome very effectively with a piece that has any combination of different insertions, deletions, inversions, different mutations. And then I'm also very excited about Vijay Ramani's technologies, an example of a suite of technologies that are coming out right now that are taking single cell genomics, where we're seeing the average of the maternal and paternal chromosomes in each cell, to single molecule genomics, where if there are different genetic variants on the two haplotypes, we can read that out and also look at epigenetic measurements on the same single molecules, such as chromatin accessibility or gene expression. So these two technologies paired with sequence-based deep learning for interpreting human variants is a really exciting frontier that we're working towards in my group and in the Institute with our collaborators. And I'm going to focus the rest of the talk on explaining how we do this close experimental and computational collaboration. So what that looks like is we start with data that's available from public databases, from experiments that we've conducted here, and whatever technology exists right now, and build machine learning models that take sequence as input and predict some activity of that sequence. So very generically, the input could be a DNA or a protein sequence. When the input is a protein sequence, you could do things like fold the protein or predict its structure. There's many tools that everyone's aware of now that have been doing that for a number of years. What we work on primarily in my group are DNA models, so models where we take a swath of the genome as input and then we predict different kinds of activities of that sequence, things that can be measured genomically or maybe through other technologies. We have a few projects on imaging-based readouts, something that can be measured about that sequence. So gene expression, the folding of the chromosomes in 3D, or epigenetic marks. Now, once you have a model that's reasonably accurate on held out data that it's never seen before, you can use it to make predictions of what happens if I change the sequence. Now, a lot of the most performant deep learning models for sort of predicting one kind of genomic measurement these days take sequence and other data as input. For example, if you want to predict gene expression using sequence around the transcription start site and epigenetic marks works much better than just providing sequence, because there's a lot of information about the cell type specificity and about the activity of the promoter in that epigenetic data. However, if you wanted to deploy that model that takes multiple different experimental inputs, if you wanted to deploy it and make a personalized prediction for a new person, you would need not only their genome sequence, but you would also need to perform all those epigenetic experiments. You would need to know how the, say, chromatin accessibility or histone modifications change. And so we're really pushing these sequence-only models, not because they're the best out there at predicting any given readout, but because they're very deployable at scale on biobanks, where we often have only whole genome sequencing for the participants. And so what you can do is then use this model to say, well, what if the genome looked like this? And put in the variants individually or in whole haplotypes on the computer. This is sort of genome editing on the computer. Put them in and they get a model-based prediction of how the activity would change. And then use that information to develop causal hypotheses. Well, it seems like when we delete these particular transcription factor binding sites, expression goes down in this locus, but it goes up in this other locus, would be an example of that. And that's because the sequence context is different. There's other things that are binding differentially in those two loci. And you can do a lot of these computer experiments. It's quite feasible to do millions or trillions of them. Each individual one doesn't need to be perfectly accurate, but as an ensemble, they provide sort of a picture of what the model thinks would happen if you perturbed the genome in certain ways. And that can help you develop really reasonable hypotheses. At this point, they're just hypotheses. They're model predictions. And so it's very important to go back into the wet lab, use the model predictions to design experiments, test some of your top hits, test some of your bottom hits too, things you think shouldn't have an effect, things with a medium score. Test some things that you as a scientist, based on your reading of the literature, think are probably not a great experiment to do, but the model puts a high score on it. Test some of those and just see. Maybe our literature knowledge and our fear of taking risks in the wet lab because each experiment is so expensive and time consuming. Maybe we're not trying things that we should be trying. Or maybe that's wrong. Maybe we're actually, our literature-based knowledge is really wise and the model is making mistakes. Either way, the data generated in this next loop around will help us make a better model in the next iteration. So this approach has been applied by us and many others to study genes and proteins, to study gene regulatory elements like enhancers and insulators and repressors. And then also, and what I'm going to use as my example today, because I think this is a bit more unique that we work on this, although there are a number of other groups that do. Understanding how the sequences that aren't sort of well-characterized regulatory elements aren't the genes themselves, those intervening sequences across the genome that contain a lot of repetitive elements, that don't contain a lot of known signatures of regulatory elements. What happens if we perturb those? And I'm going to show you some examples of how perturbing those sequences, like the blue in this picture of a topological domain, can actually change how the genome folds. And if you change how it folds, you can change the interactions between enhancers and promoters and affect gene expression. You can also affect how the genome replicates, its replication timing and fidelity, and other aspects of how cells function. So let's take a trip around my experimental computational closed loop here, but using genome folding as an example. So far I've been very high level on the talk. We're going to get a little more specific now. So an example of a piece of existing data that can be used to build a sequence to activity model is chromatin capture or Hi-C experiments. This is a way of measuring through a genomic assay the structure of different loci of a genome. It works through proximity ligation and paired end DNA sequencing. The readout is for any sequences that were nearby in the molecule, you will see them read at the two ends. They will get ligated to each other, even if they're not nearby in the genome, and then they will get read out through sequencing of the two ends of a ligated DNA molecule. By sequencing lots of different molecules across a population of cells, you get this sort of heat map that I'm showing on the right. Both axes are genomic coordinates. They represent the genomic coordinates of the folded molecule here. Red means a higher frequency of interaction between a pair of loci in the genome. So this locus and this locus interact a lot because their dot here is red. And then the blue is lower than expected interactions given the genomic distance. And what we can see is that different parts of the genome have different maps. And so the problem we set out to solve was, could we predict these maps from sequence? And this is a great machine learning problem because these are essentially pictures. And we knew that tools from computer imaging are pretty good at predicting pictures. And so we wanted to see whether we could predict that from the sequence alone. So can we fold the genome from the sequence alone? This is like protein folding just from its amino acid sequence. So the model that we developed is called Akita. This work was led by Jeff Fudenberg when he was a postdoc in my lab, and Dave Kelly, who we collaborated with, who was at Calico. Our model takes a megabase, a million base pairs of DNA as input. That goes through a convolutional neural network with a sort of typical trunk. And then the difference between this and models that predict one-dimensional features along the genome, like epigenetic marks, is that we need to average the output of the trunk into a 2D map, and then we do some more symmetric dilated convolutions on top of that, and end up with predictions like this. So you get a 1 megabase by 1 megabase predicted Hi-C map as an output. And the goal, as you're training the model, is to minimize a loss function. In our case, we use mean squared error between the data and the predictions. We train on 80% of the genome, hold out 10% for validation to do some hyperparameter tuning and to prevent overfitting, and then a completely independent test set of 10% in loci not adjacent to the loci we use for training that we can use to assess performance. And this model performed quite well. I was pretty surprised that you can fold the chromosome from just its sequence. And we got a mean squared error genome-wide on average 0.14. That's pretty good. And maybe what's easier for people to comprehend is this isn't a hand-picked excellent example. This is typical of a prediction and data. So with that model, what my lab's been doing for a number of years since we built Akita is making Akita models for different cell types, but also using those to do this disruption scoring. So to ask what happens now if I perturb the sequence. So we developed a tool called Supremo. What it does is facilitate the process of doing genome editing on the computer. So if you have a reference input and it would predict a map like this, here's a different locus with a different folding pattern, just so you can see that not every locus of the genome looks the same. And then you can provide the model with a file, like a VCF file with a bunch of variants in it. So let's say for this particular sequence, there's a deletion in the middle. Those pieces get deleted out on the computer, and then another prediction gets made. And you can see that while the structure is generally similar, there are some differences in the prediction between the edited and the unedited sequence. And we use a suite of different statistics to compute in an automated way, because we want to do this for trillions of them and not have to look at each set of pictures, compute statistics that measure how different the maps are. And we had a recent paper sort of benchmarking different statistics. Not necessarily to say that one is absolutely the best, but to highlight for other people who might want to do this, what they pick up on. Do they pick up on sort of pattern changes or intensity changes, or do they really focus in on particular kinds of features that have changed? So we can compute a change in the chromatin folding as a score and then use that. So we have applied Supremo to a bunch of different problems. I'm going to describe a few of them now for the next part of the talk. The first one is going in to several different databases and looking at whole genome sequencing for families with healthy parents and a child with a developmental disorder. We looked at congenital heart defects, or CHD, and autism spectrum disorder, or ASD. And had about an equal number of families from each disorder. And the challenge we wanted to address here is the fact that for both of these, most of these families do not have a genetic diagnosis for their child, even though these are both considered to be very genetically influenced diseases, genetically caused diseases. For CHG, 65% of the kids do not have a damaging protein coding variant in a gene that's expressed in the developing heart that could be a plausible causal protein coding mutation, and that number is 80% for autism. So there's a lot more to explain here about what's going on, and we thought maybe genome folding plays a role in some of these cases. So we applied Supremo with the model Akita, and I should say Supremo can be hooked up to any model that takes sequences input and makes different kinds of predictions. But in this case, we're hooking it up with Akita and making predictions of whether the genome folding is different in these individuals. So we look at the whole genomes of the moms, the dads, and the kids. We try to identify, we started by looking at de novo variants, sequences that have a variant in the child that was not inherited from either parent. We also have a project looking at oligogenic variant combinations, so cases where the parents passed on variants, but one came from mom and one came from dad, so the combination didn't occur in either healthy parent. But here we're just looking at, I'm just going to show you results for single de novo variants in children. I'm going to focus on deletions because those tend to have pretty profound effects on the genome folding, and they are also pretty easy to interpret what's going on. So here's the result. So the probands, this kids with developmental disorders, had more disruption, according to Supremo Akita, than healthy controls. So here's the data for developmental delay and autism from the Safari families. And in this case, we were able to compare to a healthy sibling. So the healthy siblings are in blue. The cases with autism are in the salmon color. And in both the sick kids and the healthy kids, there are some variants that Akita says, no, doesn't really affect the genome folding very much. They have low scores, and we consider low scores sort of things below about 5 on this log scale. But there are some high scoring variants in both the sick kids and in the controls. But what you can see by the sort of excess of pink on the right side here is that there's more damaging variants in the cases than in the controls. And we also have seen that they tend to be in different parts of the genome. The control variants are localized in very specific parts of the genome that we're hypothesizing tolerate variants. Whereas the case ones seem to be kind of smattered all over the genome and enriched for being nearby psychiatric disorder genes. We also saw the same thing in congenital heart defects. In fact, the signal was maybe a little bit stronger, a real shift towards higher scores in the cases compared to the controls. So we can start looking now at these high scoring variants and try to figure out what's going on. We can, with a smaller number of highly prioritized variants, we can actually look at the predicted maps and we can look at what kind of contacts are being lost, what kind are being gained. And so we saw a number of interesting patterns, but one that we saw a lot was something called enhancer hijacking. And this is just a cartoon of it. So suppose in a healthy genome, like a sibling, a locus looks like this. There's a gene with low expression in the relevant tissue. There are some strong enhancers, some good regulatory elements over here to the left that could turn that gene on. However, their chromatin interactions are going off the screen to the left. Because there's an insulating boundary here in between, bound by the protein CTCF, which is known to block the cohesin-mediated loop extrusion process and enhance, not completely prevent, but enhance chromatin interactions on one side of the other that are on the same side. So interactions to the left with each other, interactions to the right with each other, but to not completely block, but decrease the chance of interactions across the boundary. So if the patient now has a deletion of this insulating boundary, then the enhancers now are not prevented from interacting with the gene, and you could get upregulation of this gene at the wrong time or place during development. So this is just a hypothesis, but we saw many examples that look like this and made us think this is an interesting thing to go and then test in the lab. We also saw gain of insulating boundaries through other kinds of modifications to sequences, insertions, and inversions. And so the enhancer hijacking isn't only this sort of gain of interactions, it can go both ways. But focusing on this,
Yeah.
hello?
Oh, okay.
Did you have a question, TM?
ماشي. ده هي بقى ما عرفنا حركات باك. ها؟
I, I think maybe you need to
ده
mute.
نرى باك.
Okay, I'll keep going. So we then go into the lab and start testing. And when we started these experiments quite a few years ago now, we didn't have a high throughput genome editing approach. So we've only done a handful of them, but I'm excited to show the results and then to also explain how we're scaling this up. So we go into induced pluripotent stem cells, or iPSCs, we edit them, and then we can differentiate them into the correct cell type for any given disease. So we focused mostly on excitatory neurons for autism and on mesoderm and cardiac progenitors and cardiomyocytes for congenital heart defects. But one could generate other kinds of cells in the lab. We then have an isogenic comparison, meaning we have an IPS line that is not edited, and we have one that's identical, except that it carries the patient deletion. And we can perform experiments, do Hi-C and RNA-seq and other experiments on replicates of these two different conditions with and without the patient variant to see if the variant is truly causal. So here's an example. This is a patient with a de novo deletion in the MESP1-2 locus. MESP1 and 2 are important for the mesoderm stage of cardiomyocyte development and differentiation. This is the prediction of what the locus looks like with the reference genome. Here is the prediction for the deletion, and they're adjacent to the deletion are these gain of contacts, which look like an enhancer hijacking of this gene KIF7, which is a disease gene for ciliopathies, which are a type of heart congenital, one of the things that contributes to congenital heart defects. So this looked like an interesting one to pursue. From the predictions with Benoit Bruneau's lab, Jingxing Wu, we did the experiment. So here is the Hi-C or capture-C in this case, meaning focus just on this locus, data at the mesoderm stage. And we see for the unedited cell line, it's quite blue here. And we see a really strong gain of interactions, maybe even stronger than what the model predicted when we do the deletion in this cell line. So the Akita model prediction was very accurate. We then went on and did RNA-seq, and we saw that there were hundreds of genes differentially expressed, including in this locus, but also genome-wide, suggesting that perturbation of the expression of the genes locally here might be having downstream effects. To show that that is actually the causal pathway and to look at this outside of cell lines and in an animal model, the Bruneau lab now is making mice with both heterozygous and homozygous copies of this patient variant. And so this has led to a whole new, research direction in our collaboration based on this machine learning prediction. Here's an example from autism spectrum disorder. This locus was from a patient without any plausible explanation for their disease with a 22 kilobase de novo deletion on chromosome 10. This locus has a large number, 4, with the stars next to them, behavior-associated genes that are already in various ways been linked to neurodevelopment and to different types of behavior. And so this looked really interesting. We had a reference prediction and then an alternate prediction where we had both gains and losses of contact. So here's a loss, here's a gain of contact. We were also intrigued because one of the genes here that loses contact is expressed in neurodevelopment according to brain span, and it is downregulated, so differentially expressed in the correct direction in the brains of individuals with autism spectrum disorder compared to healthy controls from the psych encode cohort. So again, we did the experiment. Now I'm going to show here it's in iPS-derived excitatory neurons, and I'm going to show a little bit different visualization here. But rather than showing the two maps, I'm going to show the difference between them. So, and I've also taken the map, I've taken the upper triangle of the map and then rotated and flipped it on its side. So sorry for the change of orientation in the presentation, but I think these difference maps can be really helpful to see the differences, the changes. And they also enable us to plot things in a genomic track below the Hi-C map. So I really like that. What we see here is both decreased and increased interactions consistent with Akita's prediction. So this is the experimental unedited versus edited cell line. These look like what Akita predicted. So again, we validated the model prediction. And we see some in normal cell line here, some high chip data and some CTCF. For CTCF, we can see that there are some chromatin loops that would be affected. So that's sort of consistent with that enhancer hijacking theory. And then we see where the different genes are so we can hook them up with what's going on in this locus. And this work was done with Yin Shen's lab at UCSF. So again, we validated this has led to some new plausible candidate disease genes and a mechanism that's not mutation of the protein, but misregulation of the protein through disrupted genome folding. The third example I want to show you is from pediatric brain tumors. So we had a project where we have been scoring all of the variants from the cancer, the CBTN, pediatric brain tumor database, and collaborated with the Damande Lab at Weill Cornell and the Kids First initiative, which is a collection of developmental and cancer variants for diseases that children get. And so we acquired all of these tumor genomes, and we identified variants, somatic variants in the tumors that weren't in the germline, and then we scored them again with Supremo and Akita. Here I'm showing the results in a different visualization. This looks sort of like a Manhattan plot from a GWAS because what I'm showing is the chromosomes along the x-axis. Um, and I'm showing two different scores, our correlation score and our mean squared error score, just to show you that they don't pick up on exactly the same thing. What you see here now is all the variants from a large cohort of tumors. So each tumor has many variants, and then these are pooled here across all of the kids. And I'm doing that here to emphasize that there are some loci here, two on chromosome X, one on chromosome 19, one on 7, and one on chromosome 3, where there is recurrent prediction of disruption. So independent variants and independent children are getting very high scores in these exact same 1 megabase windows of the genome. These are not mutated more in the tumors compared to other loci. So these aren't mutation hotspots. These look as mutated on, if you were just looking at the mutation rate, as mutated as other 1 megabase windows. But according to our machine learning model, the mutations that occur there are having an outsized impact on genome folding.
Katie, can I ask one thing? Yeah.
Yes.
Those variants now, are they structural variants as well, or are they?
Yes, we scored all the point mutations as well as all the structural variants. These included deletions, insertions, inversions, and then also the complex break ends. So like the more complex variants where you get chromosome fusions and you have to look at them in terms of all of their break ends and you know the complicated variant calling that comes along with that. In fact, I'm showing some here, just one example of many that occur in each of these recurrent loci. And what these maps look, the way these maps look indicate to me, and that may not be obvious to you, but they indicate to me that there are different kinds of variants being displayed here because the whited out bits are sometimes on the alt and sometimes on the ref. So when it's white on the ref and there's color here on the alt, that's like an insertion, right? And when it's colored on the ref but masked out on the alt, that's because of a deletion. And then these ones that look, actually many of these here are complex variants with two chromosomes coming together. Yeah. And so what you can see just through these examples is that there are some pretty dramatic, these high scores up here also visually correspond to dramatic changes in the maps that we pick up on these difference maps at the bottom. So I thought this was really interesting. It points us to some loci, some of which contain known cancer genes, some of them not as much, but point either to new loci or to maybe a new genetic mechanism in loci with known cancer genes. The last thing I wanted to talk about now is a more unbiased approach. So far what I've presented is going in to patients with really devastating conditions and looking at some pretty dramatic genetic variants like these complex structural variants. But one with these machine learning models can also just make any mutation. We don't have to have ever seen it in a person. We can just ask the model, what happens if I make this change in the genome? So this work took the entire human genome and performed deletions of every 5 kilobase. So every little gray dot here is a 5 kilobase window that we deleted, and we performed the Supremo Akita analysis and computed a disruption score. And then the blue curve is just a moving average along here, a smoothed curve to show that there are regions of the genome that tend to have higher disruption on average, and some that tend, like here in the middle, to have quite low disruption on average. And I'm showing a big swath of chromosome 7 as an example, but we went genome-wide and did this. And what this enables us to do, it's completely unbiased. We just move along and delete all the windows. But afterwards, we can ask, well, what's going on in the windows with high scores versus low scores? So we have a publication that talks about a lot of the different correlates of high and low scores. I'm just showing a few here. One of them, as expected, the high scores tend to have more CTCF binding. In general, they're more gene dense. So the model predicts more disruption in these gene dense loci compared to the gene poor ones here. But a really interesting and perhaps a bit surprising correlation, one of the strongest ones we found, were with a type of repetitive element called ALUs. There were a lot of these ALU elements in the high scoring regions. And so this wasn't unique just to the ALUs. The ALUs were the strongest hit. But other kinds of repetitive elements were also enriched in these high scoring loci. So we developed this hypothesis that they may actually play a causal role in genome folding. This is a busy plot, but I want to show what we did next in detail. So we then, after doing this unbiased thing of 5 kilobase windows and developing this hypothesis that the repetitive elements could be important, we said, well, okay, let's go back and just delete the repetitive elements specifically. And in fact, we're doing experiments now where we just delete parts of them, and we had a little bit of that work in this paper too. So up here are just random deletions of the same average size of these repetitive elements. And then these are the different classes of repetitive elements. And we deleted many, not all of the instances, but a large random subset of instances from each of these families. So for example, here are the hat elements, and some of them have low disruption scores, some of them have high, and this shows where on average hats sit. Now, we also, as a positive control, deleted all CTCF binding sites. And you can see where that sits. So these sort of size-matched regions with at least one CTCF motif in them score pretty highly on average. But there are a number of classes of repetitive elements, and in particular, these ALUs actually score higher than CTCF. So the model is attending to them and changing its predictions more for these repetitive elements than even for CTCF, which is known to play a major role in genome folding. We then thought, well, maybe these things are mimicking, maybe they have sequences in them that look like CTCF binding sites. So over here, what I'm showing in orange and red is that for the ALUs and all these other ones, only a small proportion of the high scoring ALUs have a CTCF motif in them. So overall, this can't be explained by having CTCF motifs. There are some other sequences that have a bit more of the CTCF motif, but in general, amongst the top, quantile, or 10%, I think it is, decile, of high scoring elements in each of these categories. On the right, what we see is that most of them do not contain CTCF, and many of them are also not transcribed. And that's important because transcription itself can create a chromatin boundary. And so this suggests some other causal role for these, and launched a whole series of different experiments that we're doing to follow up on this, both computational as well as experimental. So to conclude, the Akita model predicts effects of variants, including patient variants, on chromatin folding. It does that highly accurately and suggests that genome folding may play a role in developmental disorders and in cancers because we see elevated disruption scores. And these childhood brain tumors have these hotspots of disrupted genome folding that are not mutational hotspots. We've done genome editing to validate the genome folding, and that we also saw expression changes. I showed you a couple of examples of that. We've done a handful now. So far, all of the Akita predictions have been correct. It's not enough for me as a statistician to put a p-value on that, but so far the model has been really accurate as to what's actually happening in cells when we insert the variants. And we're now gearing up to do this in a more high throughput way, so we could put a p-value on that. And then this in silico mutagenesis can be used beyond patient variants to discover novel sequence determinants of chromatin contacts. And I would emphasize here, I use the chromatin folding as an example, but everything I just said could be done for a model that predicts RNA as a track or predicts epigenetic marks or chromatin accessibility. And we're working on all of those now. So the sequence to X with the genome folding can be used across all of these different aspects of the genome. And I just want to briefly talk about what we're doing to scale up now. So the idea is to train models with the data that we have now, make predictions like I just showed you for the effects of variants in different cell types. So we've trained models that make predictions that are unique to different cell types. I'm showing here pluripotent cells, excitatory neurons, and cardiomyocytes. We're also working on various cell types relevant to cancer. And then we would come up with a bunch of in silico experiments that help us design the experiments we would want to do in the wet lab, these perturbative variants through the in silico mutagenesis or Supremo process would help us come up with haplotypes that we would want to then edit into cells. And as I mentioned earlier in the talk, there we're expecting to be able to test hundreds, maybe thousands of different haplotypes because this technology called Editrons from Seth Shipman's lab makes it pretty easy to go in and replace a whole chunk of several kilobases of the genome with a piece that either carries or doesn't carry a variety of different variants in it. And we do that using this retron that synthesizes the repair template hooked up to the Cas protein as shown here. So we take the IPS, the pluripotent cells, we edit them, we differentiate the edited cells, and then we're going to read out the effects with a variety of single molecule as well as bulk assays. So we're going to look at things in bulk, but I'm really excited about this single molecule work. I'll highlight Samosa specifically, but the Ramani lab has a number of different really exciting single molecule assays, including one called LASSI, which basically does open chromatin and high-C at the same time on single molecules. Samosa measures chromatin accessibility or nucleosome depletion, and what you get are these, for each molecule, a picture of the footprints across it. And what's really neat in terms of making this high throughput, anyone who does any genome editing or works with people who do may know that a really rate limiting step is after you edit cells, you have to clone out. You have to find a clone that actually has the correct edit in it, doesn't have additional incorrect variants that you didn't want to make or isn't unedited. And then you have to expand that clone, and that process of cloning and PCR is the main rate limiting step in this whole thing. With this single molecule readout, we can read on a long PacBio or nanopore sequence of many kilobases long. We can read out the edits that were actually made, and we can read out the effect of them on the chromatin accessibility without any cloning. So this is a pooled assay. You put a whole bunch of these constructs into cells. It's sort of like Perturb-seq, where the readout is single cell RNA. Now we're reading single molecule chromatin accessibility as a readout, and we don't need any barcodes. There's no cloning. We just read out on the molecule itself, whether it was edited or not. And so if we get a bunch of molecules for a given locus, and some of them are edited and some are not, we can now do statistics and try to compute whether the variants are having an effect on with Samosa chromatin accessibility, but we can also look at chromatin interactions and gene expression. And we're exploring some sort of droplet-based systems to sort of do these as a multiplex. So I'm really excited. It's early days, and I'm sad that I don't have anything more than to tell you that we have preliminary results showing this works. But I think very soon we will be able to scale this up and get our first round of data that we would then use iteratively to improve the model and continue around the cycle. And so this is really an engineering perspective that I'm trying to share today, sort of a design, build, test. We're not claiming every model is perfect or that it needs to be perfect. What I think the value add is it is that we can go around this circle quickly and iteratively, and improve both the wet lab and the computational approaches as we go. And accelerate the rate of knowledge generation about what's causing genetic diseases. And as I highlighted earlier in the talk, also use it to build better therapeutics. So with that, I will thank our funding sources, the amazing folks who did the work and who collaborated with us, and I'm happy to take questions.